The LatinX community is an affinity group at the Broad with the mission to bring people together and empower them to reach their full potential. At this very moment, we need people in the steering committee to help us achieve these goals. So if you're interested, please reach out to us, as we'll provide links and comments to do so in the future. Following this mission, we created the LatinX speaker series with the purpose of identifying and highlighting Latinx and diverse researchers within the Broad Institute and abroad. We want to foster collaboration, and we want to allow visibility for role models for diverse individuals. We also wanted to provide opportunities to network and to promote research within the Latinx community. And with this out of the way, we'd like to move to our speaker today. And our speaker is Dr. Erica Vieira. And Dr. Vieira has conducted basic and translational research in neuroimmunology over the past 10 years. She has focused her research on evaluating peripheral biomarkers in psychiatry and neurological disorders to identify the link between the periphery and central nervous system, and with the main focus as an independent research scientist at the Center for Addiction and Mental Health. And we'd also like to investigate the role of, or she would like to investigate the role peripheral biomarkers play in several mental illnesses, including neurodegenerative, cognitive dysfunction, and mood disorders. Her long-term career goal is to establish a sustainable, diverse, and independent laboratory in Canada. And after completing her master's and her PhD in the Federal University of Minas Gerais in biochemistry and immunology, she had the opportunity to investigate the role of immunological changes in different neuropsychiatric disorders across the lifespan, including major depression, bipolar disorder, Alzheimer's, and dementia. So take it away, Dr. Vieira.
Thank you, Ricardo. Thank you for all committee for the invitation. It's a pleasure to be here to share a little bit of my research and the pathway that I have been developed over the past. Oh my gosh, it's never good to say how many years, but almost 15 years from now. So it's a pleasure to be here, and please feel free to contact me. And I know that after my presentation we have some Q&A questions, so don't hesitate to contact me. So today I will talk about the liquid brain biopsy, the brain-derived extracellular vesicles in neuropsychiatric disorders. This work actually started quite early on my career, the past 3 years. So this is one of the main focus that I have on my lab. And I will try to explain for you how these extracellular vesicles just come into my life. But first, I have no disclosures for this presentation. I would like to tell you about who I am and where I come from. So as Ricardo said, I did my PhD in immunology, my master in biochemistry, and my postdoctoral fellow for 7 years in neuroscience. One thing that I knew it since the beginning is that I like to collaborate. And to do research, especially in the country that I come from Brazil, we need to support each other. So this network, be a networker and a collaborative independent person helps me to achieve the goals and see more clearly where I want to go with my research. The third thing that I'm super proud of is that I'm the first woman in my family to go to university, my sister and I. So I always try to encourage not only women, but researchers from underrepresented minorities to pursue a scientific career. Especially in academia, it's not easy. So have those support is something that I always willing to do for the researchers. And the thing that I love most on my work is that I'm completely in love with science. Since I was an undergrad student, I'm still sometimes late in my lab, process the data, and when I plot the graphic and I saw something that is really excited, this made me laugh. So I hope that everybody could be able to do something that they really love to do. So I'm glad to be one of those persons that can certainly say I'm in love with science, and this is something that make this really hard pathway much easier. So always be in love with something that you do. And as a Brazilian, sometimes we think that the obvious is sometimes not so obvious as we think about. And I want to bring a fact here. As a Brazilian, of course, that I love football. And in 1997, we have this goal from Roberto Carlos. And it was obvious that he was not able to score the goal. But sometimes the obvious is not so always obvious. And let me play this goal for you. So he was very far from the goal, and he takes like a very long distance, and then we can see the kick. If you see, everybody stopped at the moment, and the curve, look, everybody thought, no, this ball is going to the wrong direction, not to the goal, and he scored. But as a scientist, the impossible free kick from Roberto Carlos has a physical explanation. So he worked with high pressure and low pressure in the ball, and he scored a goal that was quite seemed like impossible for much of those athletes. And we have the score was 1-1 against France. But anyway, so I remember I was a teenager at the time when I saw this goal, and this is awesome. And science is sometimes it's like this. We need to put our faith and our acknowledgement in something that sometimes we don't know if we will achieve our goal. But we always need to think about the impossible, 3 impossible things every day. And one of the impossible things that I was trying to work with, I always had this huge experience with peripheral markers from different inflammation, metabolic, neurodegenerative proteins. And I was wondering, how can I connect those two? Every time that I submit a paper, I got this feedback. How can you guarantee that the measures that you have in plasma or a cell culture or anything can really reflect what is going on in the brain? Especially because we like to compartmentalize everything. So we think that it's 2 things different. So we have the brain and we have the periphery, but they quite often talk with each other all the time, actually. And we can see from preclinical studies that this translation, this communication between periphery and central nervous system can really affect the outcomes for some neuropsychiatric disorders. So I always thought that I worked to build this bridge between central nervous system and periphery, but how could I connect those two in a more assertive way? So this is why I start to work with extracellular vesicles. And to prove my concept, I start to work with 2 cohorts. So one of the cohorts that I had access was the SENDEP cohort. The SENDEP is the link molecular senescence change to depression and cognitive impairment in late life. So we recruit, is a longitudinal Canadian cohort where we recruit patients without depression and with late-life depression. And it's an observational study, and we follow those old adults for 5 years. So we have neuropsychiatric assessments and also blood and MRI in 113 old adults. Unfortunately, because COVID, we still struggle to have a linear pathway for this cohort, but I will present for you anyway the preliminary results that I have. And the second cohort that I had access was the standardizing care in neuropsychiatric symptoms and quality of life in dementia, STEM. So for the STEM, we work with Alzheimer's dementia and nursing care homes. So all of those individuals, they have dementia in a very advanced stage. So I have 2 points. I have the late life and the dementia. And for those who may know, depression could be a prodrome state or a risk factor for dementia. So I was wondering, can we have a better way to evaluate peripheral biomarkers in those two cohorts and maybe come up with some biomarkers that could really help us with prediction and outcome? So the first thing that I did, using a panel for 356 different proteins, I measured the proteins level for different class of biomarkers. Here, I'm just brought some of them and I divide them like for inflammation, endothelial risk factor. And here I'm present to you the fold increase over control for the patients with late-flat depression. And all those measures were made in plasma sample, okay? So the question is depression a prodrome or a consequence or risk factor for dementia? And here we have the level. The red line is the one that is the same ratio between controlled and depressive patients with depression. And as we can see, we have some proteins that we have not only above, but below these threshold. However, when I overlap the patients with dementia, we can see that the profile is not the same. For some of them have a huge increase of those proteins in plasma sample. For some of them, they are almost the same or even below the late-flat depression. Okay, so inflammation is important core for depression and also dementia. But can I see the same behavior in different class of protein? And my answer for you is yes. So I stratified, I just brought some of them. So here we have the chemokines, neurotrophic factors, metabolic proteins and metalloproteinase proteins, and also endothelial vascular proteins. And as we can see, we have the same profile. So for example, look, BDNF for patients with Alzheimer's dementia, and also PGGF, both isoforms, AA and CC. And the same for endothelial vascular protein. So we have EGF and endothelium 1, a high fold increase over controls for patients with Alzheimer. And the same with metabolic and myeloperoxidase protein, so especially MMP1 and 9. So how can we connect what is happened and what is the shift or trigger point that make a person struggle with depression, start myocognitive impairment or a dementia process? And how can we have those biosignatures? And I just want to remember you that all of those measures were in plasma sample. So when we think about inflammation and cellular communication, when we go to the in the books, we have this process. We have first the presentation of the antigen from an APC cell, and then we start the transcription and the traduction process. However, the cellular communication is a process very, very dynamic. And as we can see, this is a view where we can see dendritic cells in green and B cells in red. And the same dendritic cells can activate at the same time with different epitopes of the antigen epitopes. So many B cells is a very dynamic cell. In less than 5 minutes, we already have an FKB important factor that goes to the cytoplasm, to nucleus, and start the RNA, the messenger RNA for inflammatory cytokines. So this is a very close and dynamic process and happens all the time. However, when we think about neurodegenerative disorders, when this process starts, it's quite hard. And using human samples, human subjects, have the genesis or the beginning of the process, sometimes it's really hard to track those things. So now we are looking for biosignatures and how can we connect what happened in the central nervous system, the periphery, that could really help us using different algorithms and artificial intelligence, and tell us how can we use this information to predict the outcome. So one of the things that I'm trying to do here is evaluate the circulating extracellular vesicles. So the EV, they function as a mediator in intercellular communication by transfer biological information between the cells. Sometimes I like to compare when the students say, oh, I never heard about extracellular vesicles. I like to compare them like a Wi-Fi signal compared to a cable signal. So that is how can I say, it's not about be essential, but be a very effective way to send to another cell the message that a cell far from them. And it's also a very efficient way to deliver the cell. So the extracellular vesicles, we are talking about membrane. So this membrane can attach the target cells and delivery right away the message inside the extracellular vesicles. The extracellular vesicles is a family of vesicles that range from 30 to more than 300 nanometers. But usually we divide these extracellular vesicles family in three main categories. That is the exosomes. They range from 30 to 150 nanometers. We have the microvesicles, 150 to 600 nanometers. And above 600, usually apoptotic bodies. So the EV can be released under normal and stress conditions by any different types of cells, including cells such as neurons, microglia, oligodendrocytes, and astrocytes. So using immune capturate, I'm trying to immune capturate the extracellular vesicles and the circulating extracellular vesicles using two antibodies. That is a LU1CAM, that's for neurons, the orange enriched EV, and the GLAST, that is a glutamate aspartate transporter 1 for astrocytes. So as I said before, in the healthy central nervous system, the cells, they exchange EV all the time to keep the function, the homeostasis. So for example, oligodendrocytes, they release EV with neurotransmitters to the neuron. The neuron also releases extracellular vesicles to keep the best the homeostasis with microglia and also support astrocyte function. So this happens, a process that happens in the homeostasis. However, what we saw, especially with preclinical and in vitro studies, is that when we induce stress, not only in the neurons, but also in the glia cells, we change the frequency of extracellular vesicles and also the message inside those vesicles, the content of the extracellular vesicles. So one study, they show that neurons under stress condition, they release extracellular vesicles with amyloid and also p-tau protein. For those that may not know, but those are the two main proteins related to dementia, especially Alzheimer's dementia. So the plates, the amyloid plates, are one of the pathophysiological process more established for dementia. And the microglia and the astrocytes, they increase also the number, and the content of the extracellular vesicles now is enriched in heat shock protein and also pro-inflammatory proteins, especially IL-1 beta. So we change the environment. And the extracellular vesicles, as I said, is a very efficient way to deliver the message. And because of their very tiny vesicles, they can cross the blood-brain blood-brain barrier quite easily. And we can measure not only in plasma, but also in urine, in different biofluids. So the International Society of Extracellular Vesicles, they published a guideline in 2018, and they will publish 2022, hopefully by the end of this year, the update of this guideline of how can we really guarantee that we are looking for extracellular vesicles and not debris. So because they are so tiny, we have so many factors that can interfere in our analysis. So the last decade, as they published, the last decade has been a sharp increase of number of scientific publications describe physiological and pathological functions of extracellular vesicles. However, specific issues arise when work with those entities, whose size and amount often make them difficult to obtain in relative pure presentation and to characterize properly. And this is the first barrier that I had working with extracellular vesicles. So before you start everything and work in a hospital, people sometimes they heard about extracellular vesicles and they call me, oh, I have 300 samples, I have 100 samples, how can we evaluate extracellular vesicles? So to move on with my analysis, I decided to work with flow cytometer. So we have a special type of flow cytometer that is not photo multiplicator, but is a photoiodide. It's a different way to measure the sample with more sensibility. However, so first I need to have the machine. Secondly, I need to establish a pre-analytical phase. So I work with blood collecting citrate, sodium citrate tubes, and I remove the platelets interference on my samples. Most of the EV in circulating EV come from platelets. In my case, I don't want to work with platelets. I want to see the orange of the EV. So I want to enrich my sample with EV, but not from platelets. So we do two spins to have the plasma pouring platelets and then the plasma free of platelets. And I always collect 2/3 of my sample and I dispose the 1/3 left. So, and then I make small aliquots, 100 microliters, and then I start my biorepository bank. Then I validate my equipment. So using the rainbow beads and small beads that I know the size, I was able to establish the range of accuracy from the equipment that we have here in the institute. So those were the 3 steps. And then we move on to how can I isolate the EV? Because it is a lipid, it's a vesicle, it's a lipid membrane. We also have a bunch of lipids in plasma sample. How can I guarantee that I'm looking the extracellular vesicle, not the debris or some disrupted membrane from the cells because of my blood process? So the first thing that we do, we evaluate the 100 seconds of my acquisition. That is the plot number 8. Then we move on with, I stain I have a fluorescence probe that is stain everything that is lipid. So it's like I enrich the signal for the lipid part. And we also need a special configuration of the flow cytometer. So I can select here everything that is lipid and has the fluorescence for my marker. Then using a formula equation, we transform the mean fluorescence intensity and diameter and from this population here. So as you can see, we have 2 main populations here. These are also lipid debris, but my extracellular vesicle is this small here. And how can I know? Because I use 3 different antibodies stained with the same color fecal erythrin. So I use CD9, CD81, CD63. Those integrins are the main markers for extracellular vesicles. And after I got this, how can I say, profile, I disrupt the membrane of the extracellular vesicles with the LatinX. Because if I have a vesicle, if I treat my sample with a detergent, I will completely remove this population. And this is what I saw when I do my treatment. So then we have our immune capture the extracellular vesicles using the 3 antigrins plus the glass or L1 cup. So in order to do this process, every time that I run a sample, I have close to 9 to 12 different types of control to guarantee that I'm looking the extracellular vesicles. So every solution that I use on my experiment, they go through the same flow. The probe that we use, the lipo, that is a viral particle that we use to transform the mean fluorescence intensity and diameter, the size, my cocktail, and also a positive control that is EV, purified EV from platelets. So I have all those negative and positive control every time that I run a sample. In summary, I have all events in my plasma, my fluorescence probe, plus the antibodies that I use to identify the extracellular vesicles, and then the antibodies that I want to really evaluate, that is astrocytes derivate and brain neuron derivate extracellular vesicles. So the population that I will present the results are from this area, this region. Okay, I hope that I was able to make a long story short, and I tried to do this over the past 2 years and a half, and now I can apply this method to the other cohorts that I have here in the hospital. So the first results that I present is the late life depressions from SENDAC. So our hypothesis was we have low-grade inflammation, depression. The senescence associated secretory phenotypes is high in depression. So I thought we have more extracellular vesicles. But my first results, when I count the total extracellular vesicles, the number is that I have less extracellular vesicles than compared to control. And I was disappointed, but it is what it is. We cannot change our results. And when I stratified for neuron derivate and astrocyte derivate, I had low levels in neuron derivate extracellular vesicles. And let's see in Alzheimer, no difference between the total number of extracellular vesicles. However, when I move on for neuron and astrocytes, I saw increase in the total extracellular vesicles from astrocytes. No difference between controls and AD for neurons. I just want to keep, it's important to keep in mind that the patients, they were matched by sex, gender, and also for age and educational level, because most of the assessments, the educational level is also important, okay? But using flow cytometry, we can see the diameter in nanometers. So I split my population in, I have extracellular vesicles, but how many of those are exosomes and microvesicles? How the exosomes and the microvesicles are generated is different. So this is one of the main reasons. And also the content of the exosome is different from the microvesicles. So I split in these two extracellular vesicles, so based on size. And what I saw is that in exosomes, now from neurons and astrocytes, is the population compromising late life depression, not the microvesicles, only the very tiny ones. And the same, but now for astrocytes only for Alzheimer's disease, in the opposite direction. So the exosomes from Alzheimer's disease derivate from astrocytes, we have 2 times more compared to the controls. So, okay, the process, one condition could be a risk factor in a proton, but they are not working at the same way. So we have a coordinated response when we think about the exosome, especially about the cell. So the astrocyte has an important role. Unfortunately, I don't have data enough to support what these exosomes from astrocytes they are delivery or how they are important in the periphery, especially. But this is something that I'm doing. I hope to have more results soon. So the second thing that I did, okay, so the exosomes, they were compromised in late-life depression and in Alzheimer. What I did, I purified the exosomes using a size exclusion chromatography. I developed a protocol to disrupt the membrane and release the protein in the micro RNA inside the exosomes. And here, I just want to show I did like a serious dilution. And as we can see, I measured those two proteins. The NDRG2 is an important protein from astrocyte function and neurofilament light for neuron, and also these proteins correlate with risk factor for dementia. So in plasma level, those are the measures, and as expected, patients with Alzheimer, they have more neurofilament light. But when I didn't treat my sample with the buffer, I was not able to measure the proteins inside the extracellular vesicles, the exosomes. However, when I treat my exosomes with the buffer, my lyse buffer, now I was able to, measure both NDRG2 and the neurofilament light in Alzheimer and late-life depression. So I just move on with this this method, and then I compare, because one of the things that I really want to do is how can the exosomes reflect what we have in the plasma sample. So the first graphic is always what I measure in the plasma sample, and the graphic below in the exosomes only. So I have here 49, close to 49 proteins. So this is like a pilot study. Now we have close to 300 different proteins that we measure in the exosomes and the plasma sample as well. So these results for late-life depression. So this is plasma. And when we evaluate the same profile for the exosomes, as expected, we don't have some measures in the exosomes for inside the exosomes. However, some proteins like CXCL2 and also GDNF, the glial-derived neurotrophic factor, they were like a glad surprise. So we have some proteins also. And I did the same for Alzheimer's disease. So this is the profile for plasma. And when we look the profile for extracellular, for exosomes, we have an overlap in some of them. But some proteins I was able only to detect in the extracellular vesicles, such as like GDNF. And we have the IL-4 receptor, that the signal was higher in the exosomes compared to plasma sample. So I think we are getting close to have these biosignatures for plasmas and exosomes. And when I overlap the total exosomes from late-life depression and Alzheimer, as we can see, we have most of the proteins, I think close to 14 proteins, they have an overlap. Some of them were not able to detect in Alzheimer, but some of them were higher in Alzheimer than in depression. So we are working now with the bioinformatics department to have more prediction and outcomes with those results that we generate in the lab. And just to give you an overview, because of the number of participants, we have close now to 55 participants for late-life depression. This is why we did the correlation analysis with the biomarkers. This is the profile that we have for plasma. So controls in late-life depression, they are quite similar when we look those two plots. However, when we go to the exosomes, the profile is not the same. So maybe when we are seeing the exosomes, they can give us a more precise answer about what is actually happen in the central nervous system. Yeah. So just to conclude, the preliminary conclusions that I have from both cohorts, and now we are extend our analysis and increase not only for different proteins, but increase the number of participants. We know that the brain cell-derived exosomes, they are vital for signal carrying in the central nervous system, and we can measure this in the circulation in plasma sample. So by carrying bioactive molecule and mediating signal transmission of adjacent and distant cells, exosomes may play a crucial role in intercellular communication, especially when we think about neurodegenerative disease. And the neural and astrocyte exosomes could also influence not only neurodegenerative molecules, but trophic stimulus, inflammation, and all other micro environmental factors. And they can be a potential window to understand what happened in this communication between the central nervous system and the periphery. Now we have a cohort with minor cognitive impairment only, without depression and without dementia. So it's like I'm adding another point on my experiment, so we can have the controls, late-life depression, MCI, and dementia. Let's see how this group, these participants, they will, the exosomes at the plasma level, if they can really support, especially the trajectory of those three neurodegenerative disorders. And we are doing some transcriptome analysis for microRNA, some microRNAs in the exosomes as well. So I want to first acknowledge all the amazing research that I work with. So Dr. Brennan Diniz and Yulia Nikolova, they are the principal investigators for SENDEX, the NIH grant. Dr. Etienne Sibyl, he's the director of the Campbell family and my boss. Dr. Tarek Raji, Sandeep Kumar, Benoit Moussin, they are the PIs for STEM and PACMD. Those are also longitudinal cohorts for dementia and microcognitive impairment. All the NIH CAMH Discovery Fund and the LABAC for support my research. The NDA group, that is a very nice group that I work close. Dr. John Dole from Sentinel Institute to help me with the validation and all step-by-step. How can we evaluate extracellular vesicles? Ana Paula Silva and my lab, the Cellular Immune Interface in Neuropsychiatry, and the Master Medicine Mark and Sarah. They are working with me close, and all of you for your time. I hope that you were able to catch, and it was, again, thank you all. It was a pleasure to present my work here.
Hey, fantastic. So the first announcement is we have a survey, so we very much value your feedback. So if you can, please post the survey, please answer the survey, and I'll send it in the chat. You also get one in your email if you registered. Okay. We also have a few questions. So the first question came from the beginning of your talk. Is the treated depression also a factor for dementia or only untreated? And I'd also like to add, if any questions come and you'd like to clarify or ask it directly to the speaker, we can also do that. So please feel free to discuss. The first question is the treated depression also a factor for dementia, or is it only untreated?
Yes, this is really hard to answer, because when we think about patients, some of the patients, they respond right away to the antidepressive treatment. Usually 1/3, 35% of the individuals, they respond quite well. However, 2/3 of the patients, they don't respond. They have they respond well in the beginning, but after a couple of months, they are in remission, they start the episode again in a couple of months. This is why sometimes it's really hard to think about medication only, because some patients, they have the course that I'm speaking about the course that we have here. Some patients, they have depression for 20, 30 years, and they don't go to MCI, like a microcognitive impairment. They stay in depression. However, some patients, they develop depression after 65 years old, and they quite quickly start with MCI and the dementia state. So I cannot say that untreatment or treatment can also develop. We have so many factors that can influence the dementia process. So I don't have a clear answer for your question based on the data that I have. And this is why those biosignatures can maybe help us because we have another factors. For example, metabolic factors also predispose to vascular impairment and also for dementia. We also have the vascular depression and the vascular dementia. So people struggle with hypertension and depression. They start this vascular impairment and they can move on for vascular dementia as well. So we have so many factors to consider, but I don't think that we can blame medication and treatment for be responsible to microcognitive impairment and probably a dementia for dementia. I don't think so.
The next question was from Dr. Joshua Levin, and he was asking for your protein data, how was it measured? Was it fluid? Like what kind of assay did you use?
Okay, so for the exosomes and the plasma, we use the LatinX platform. So we use an assay based on bead, capture beads. We didn't do like a proteomic assay using HPLC. So it was a LatinX assay, but we try to use a high sensitive molecule. And we also use the single molecule counter, the SMC, to measure very low levels of protein. So those are the two methods that we use for the protein. And for the transcriptomic, we are using a qPCR for the Micro Rna.
So the next question is from an anonymous attendee, and it says, I'm not sure I followed everything, but did you use L1 CAM to isolate neuronal exosomes? And what do you make of this preprint entitled L1 CAM is not associated with EVs in human cerebrospinal fluid or plasma? And they added the preprint, so I'm going to just post it to the chat so everyone can see.
Yes, I can see. Yeah, so this is something that I'm quite, why first, let me answer by steps. Why first I use flow cytometry? Most of the publications that we have is just with extracellular vesicles, they use ExoQuick. That is a column for immune precipitation, and then they confirm doing Western blot. So my critiques about those methodologies that we cannot count the number, and also we cannot sort the number of extracellular vesicles. So I'm not familiar with this publication, but I thank you for sharing. I definitely have a looking for. And another critique that we have is that L1CAM is a CD171 molecule. He is the most, he's the marker that most of the studies use for neural derivatives, exosomes, or extracellular vesicles. However, he's not 100% expressed by neurons. This is another factor. Some cells, including immune cells, they can also express L1CAM. So maybe this is why. So before to tell you my opinion about this article and about L1CAM is how they isolate and how they measure L1CAM. This is one of the critiques that I have when I think about extracellular vesicles. If they use ExoQuick, yes, I agree. It's an immune precipitation. It's not 100% guaranteed. They cannot quantify count the absolute number of extracellular vesicles. So I know how many exosomes or extracellular vesicles go through the laser. So I have the absolute number per microliter of sample of the participants. And what is not We cannot have this information when we think about a Western blot, but it's quite sometimes hard to measure. So this is why, yeah, so what I'm trying to do now, so why I present my results for flow cytometry, use L1CAN and astrocyte. However, I was not able to isolate L1CAN from my samples yet. So using a magnetic bead approach, I'm trying to use naked beads, conjugate naked beads with different types of antibodies from L1CAN, because it's still controversial which clone is the best one to isolate them from, especially those from neurons. So I am stem my naked bead. So first, I isolate my CD9 and 81 positive extracellular vesicles. Then I, using a naked bead, conjugate with L1CAN. So I have now 4, I'm testing 4 different L1CAN antibodies. I will try to immune capturate them and then confirm with a flow if I was succeed in this process. And I'm doing the same with the glass. But the process that I am now, I'm still struggle to find more specific antibodies to do this immune capturation. So I'm not using ExoQuick or those type of methods to isolate my neurons. But until now, I have total exosomes. I'm trying to isolate L1CAN, especially glass. I'm more confident with the astrocyte and the L1CAN. But I'm working with different biotech companies to have a better clone for the antibodies that I want to use here in the lab. I don't know if I answered your question, but feel free to contact me. It would be a pleasure.
The next question is from Gabriela Xavier. She says, great presentation, congratulations. She has a 3-part question. Are you able to recover the EVs after selection and flow cytometry? Did you also perform any other kind of characterization of EVs other than CD9 and 63 selection? And what percentage of your whole EV samples presented L1CAM and glass markers? So we can go one by one and I can go through them again.
Okay, let me see if they are those questions in the chat.
Yeah, are you able to recover the EVs after selection and flow cytometry?
I'm not doing sorting. This is one thing. So this is why I do the immune phenotype of my EV, and then I didn't do sorting for EV. I don't have instrument or a facility to do sorting. And the sorting that we have here at U of T, they don't have sensibility. The machines, it doesn't have sensibility. They don't have sensibility to sorting extracellular vesicles. So what I'm trying to do actually is a partnership with an institute to measure, to evaluate the EV and try to sorting them using the Eminis image stream X. So where they have sensibility that ranges from 30 to 40 to above 40 nanometers. The equipment that I have here, the sensibility is 60 nanometers. So first, I'm not doing sorting with my EV, so I'm just doing the immune phenotyping. How can I isolate my exosomes? It's a Psi exclusion chromatography, but I have the total exosomes, not exosomes from neuron and astrocytes yet. So what else? Presentation? Yes, I'm not doing this. Okay, the other type of characterization, we save the samples for electronic microscopy, because if we follow the guideline that I showed before from the International Society of Extracellular Vesicles, the best way to prove that you are looking for extracellular vesicles, you use like 3 different methodology. So first one I'm using EV. Second, we are isolate the exosomes for exclusion chromatography. And third, we are saving the samples for electronic microscopy or the Aminis image stream. I more tend to go with the amines because we can use the same antibodies that we use for the flow cytometer. So we can characterize and have not only the mean fluorescence density, but also the image of the extracellular vesicles. So this is why I'm more tend to, but I'm still work on those three, the other validation. And for the characterization of my extracellular vesicles, I use CD9, 63, and 81. I'm use three different integrins to validate. So usually my EV must express at least the three of those markers. Yeah. And what percentage of the whole extracellular vesicles? So as I said before, I count the extracellular vesicles. So I have close to 3,000 events. Think about the controls, the 3,000 events for total extracellular vesicles per microliter of sample. Usually is how much EV I have. For L1 and glass, they go around 1,000 to 1,050, the total amount of extracellular vesicles after my first selection.
So the next one is by Morgan Chang. So what is the size of your cohorts, LLD and AD, and are you that you are studying, and how many patients and how many controls were involved? So how big are your cohorts?
Yeah, no worry. The results that I present was 47 patients with late life depression, only with late life depression, not MCI. For controls, the number changed. For AD, I have 24, I think 24 or 27, but I think it's 24 patients with Alzheimer's disease, the results that I present. Controls match for the AD is 35, and controls met for the late life depression is, I don't record at the top of my mind, but it's close to 40 or 40-ish. But the cohorts that I have now, so the late for the SINDEP, we have the results for 113 patients with late life depression. For AD, the course that I have now, we have 56 patients with AD with Alzheimer's dementia. They could have not only Alzheimer's, but also other types of dementia. And the control, I have 65.
Any plans on scaling up more, or?
Yes, I have the results, but we are working the publication. So unfortunately, I cannot share the results right away. But yeah, so and for my cognitive impairment, MCI group is the group that I struggle more, because have the pure MCI without depression or advanced MCI is really hard to have those individuals. So this cohort is the low number that I have. I think it's 19 that I have now. And I did the same approach to see how the EV, how is the profile, and also the protein in plasma, and also inside the total exosomes. But I already have some preliminary results for NCI as well. So these are the numbers that I'm working with.
Just a quick note. So we're coming up to the hour. I'm perfectly fine to stay and answer and continue the session. We have 2 more questions left.
I can answer those, don't worry.
Yeah. So in blood, what's the approximate proportion of exosomes from the different organs, and how much of the brain versus liver?
I don't have an answer to give to you. Why? Because I have the data for what I call innate immune exosomes, because I'm using CD68. CD68 is a marker for microglia, but all cells, especially innate immune cells, they also express CD68. So I don't think that is right called microglia. I saw some studies where they call CD68 microglia, but I definitely don't agree with these.
What's the main process for handling or removing exosomes? Is it from urine excretion?
what is, okay, so the personal proportion, I have the data for brain, or for neuron, astrocytes, and innate. So I never evaluate like endothelial exosomes or liver exosomes or different types. So I don't have this answer to give to you. What is the process for handling remove exosomes? Urine, yeah. If you go to the guideline for the, I'm not familiar with the urine process because I work with plasma only. So as I said, my pre-analytical phase is collected with citrate, sodium citrate. We avoid to do to mix the tube. We always keep in vertical, and we process right away. So in less than 1 hour, we have 2 different spins in this sample, and we are super concerned to change also the centrifuge. We always keep the same centrifuge, the same spin, and everything. So this is the main process that I do for pre-analytical phase. And I'm sorry, I'm not familiar with the urine process, but I think it's probably quite similar. I can share one of the, we have a researcher, she worked with cancer and urine extracellular vesicles in BC, the University of British Columbia. I can forward to you her contact if you want to talk more about the EV from urine.
And then can you share the special cell sorter you used?
I didn't use a cell sorting. This is why I did a immune phenotype for the neuron and glass. I would be more than happy if I could do a sorting, but unfortunately I was not able to do. So this is why I'm struggling with. So together with a biotech company, I'm trying to isolate the CD9, 80, 81, and 63, and then using naked beads combined with my antibodies, I'm trying to sort via magnetic sorting. But unfortunately, I don't have. I know that we have some new, I went to the CITO conference in Philadelphia this year. We have some special flow cytometers from Beckman Coulter where they are trying to do sorting for EV, but it's the first generation, is not 100% guarantee. So I think we have something coming soon for sure.
The last questions from me. So could you share anything about the microRNA? I was very curious what kind of microRNA you were detecting in the vesicles.
Yeah, so I'm early career scientist. So every early career can tell you our main problem is budget. So because of a budget problem, we are evaluating 5 different microRNA, and those microRNA, they were selected based on Alzheimer and depression studies. So this is why we move on only with 5. But yeah, we're still doing the analysis, Ricardo. Unfortunately, I cannot share the data with you already. But we are, so the same thing, we are measure microRNA in the total plasma and in the total exosomes. It's always, I want to keep you in mind that I'm working, unfortunately, now with total exosomes. I'm not using those immune precipitation techniques. I'm trying to find a better way to guarantee and keep the same standard that the flow that I use. But hopefully, I will be able to share with you soon.
Very soon, eh? And I hopefully we'll see a biomarker from it, right?
Yeah, I hope.
All right. Once again, thank you very much for coming to talk to us here, and you did a fantastic talk. So
thank you.
Have a good rest of your evening.
Oh, thank you. Thank you all. It was a pleasure. And please reach out. Reach out me anytime. It'll be a pleasure to answer any additional questions.